Welcome to this next video in which we are discussing tuberculosis and anti-tuberculosis drugs. Okay, so what point of the story have we got to now? So we've got to the inflammatory response, and we've discussed that the acute inflammatory response is not going to be able to control Mycobacterium tuberculosis. All the acute inflammatory response does is it brings in a huge amount of phagocytes into the affected area of the lung, and these phagocytes are not capable of controlling the bacterial population, not on their own. Indeed, by the end of the previous video, we had got to this state where the rate of production of new Mycobacterium tuberculosis cells was greater than the rate at which we were killing them. So that's unstable. That means the population is going to gradually go up and up and up. So this is not looking good at the moment. The resistance of Mycobacterium tuberculosis to being killed by phagocytes is causing a big problem, and it means it's getting the better of our innate immune system mechanisms. So now, how are we actually going to bring this under control? Well, we need an adaptive immune response, and that's what I'm going to discuss with you now. So we're going to have a look now at the adaptive immune response, and the type of adaptive immune response that's going to be initiated against Mycobacterium tuberculosis is a T helper 1 mediated response, which is what I'm going to now discuss with you. So I'm going to tell you the story of how we initiate a T helper 1 mediated adaptive immune response, and then what it's actually going to do to help us bring this under control and bring the rate of production equal to the rate of killing, or rather bring the rate of killing up to the rate of production, so that at least we have an equilibrium where things are not getting worse. And that's actually the best that you ever do with tuberculosis. You bring the rate at which you are killing Mycobacterium tuberculosis cells up to the rate at which they're being produced, and then you just keep them contained in a structure known as a granuloma, and that's then what the latent tuberculosis infection is, where you've got the TB still there, but under control. Okay, right, so let's have a look at the adaptive immune response then. So, initially what we need to discuss is the initiation of an adaptive immune response. And to initiate an adaptive immune response, and actually I'll just write down quickly, the type of adaptive immune response that we're going to initiate is a T helper 1 mediated adaptive immune response, because there are lots of different types of adaptive immune responses. We're not going to go through all the different types of adaptive immune responses. We're going to go through the major type that is initiated in response to Mycobacterium tuberculosis, which is the T helper 1 mediated response. And this is a T-cell response, which is quite nice because it's simpler than B-cell responses, which you have to have T-cell responses in order to get, so it means that the discussion is a lot quicker and a lot simpler. Okay, so in order to activate T-cells then, you have to begin with antigen presentation. So what has to happen is antigen presenting cells, so macrophages and dendritic cells, principally dendritic cells, need to phagocytose the pathogen, break up the pathogen's proteins and present fragments of those on their cell surface. So firstly, before we even begin the discussion of the adaptive immune system, let me just make sure that everyone is familiar with the concept of an antigen, because this is a really important word that I will be using a lot. So an antigen is any protein molecule that is capable of having an adaptive immune response initiated against it. So Mycobacterium tuberculosis cells will have a great repertoire of antigens. They'll have a huge number of different proteins which could potentially have adaptive immune responses initiated against them. Now, these don't necessarily need to be proteins that are on the surface of the Mycobacterium tuberculosis. They might be proteins that are inside the Mycobacterium tuberculosis. So let me just draw a few of these proteins just as little blobs here. I'll just colour them in to indicate that they may be different proteins. So there's one in red, here's one in blue, and here's one in green. So all of these proteins associated with the Mycobacterium tuberculosis bacterial cell, these are all proteins that you could have adaptive immune responses initiated against, therefore they are antigens. Now, when you initiate a T T-cell mediated response against an antigen. You don't actually initiate it against the full antigen, you initiate it against a fragment of the antigen. So T cells of the adaptive immune response are not directed, they're not tailored against the antigen, they're tailored against a fragment of the antigen. Okay, and for this reason, to activate them, you have to firstly break down antigens into little fragments and present fragments on the surface of an antigen presenting cell, which can then go off and activate T cells. So let me tell you the story of what's going to happen. So Mycobacterium tuberculosis cells, they'll have a huge number of proteins that are unique to them, proteins that no human cell would ever make. And these proteins we can classify as antigens. They're proteins that could have adaptive immune responses initiated against them. What's going to happen is occasionally an antigen presenting cell, and I'll write down this keyword, so an antigen presenting cell, an APC, and in full this stands for antigen presenting cell. Occasionally these antigen presenting cells, which are all phagocytes, will occasionally get the better of a Mycobacterium tuberculosis and will be able to break it down, and will be able to break down its antigens into little fragments. So let me just discuss the examples of antigen presenting cells. So antigen presenting cells are cells that are capable of activating T cells. Now the two major types of antigen presenting cells are macrophages, which we've met, and more importantly dendritic cells. So the major type of antigen presenting cell we haven't yet discussed in this video, but you'll probably be aware of from your basic knowledge of immunology, which is dendritic cells. And let me just introduce you to the dendritic cells that you will have present in lung tissue. So dendritic cells are not present inside the alveolar spaces in the same way that the alveolar macrophages are present. However, they are underneath the pneumocytes of the alveoli. So let me just show this here. So we could have a dendritic cell here, and actually they have their little processes extending through the surface of the alveolus, like so. So I'm just showing a dendritic cell here. And the reason they're called dendritic cells is because they have these dendrite processes coming off them. They're nothing to do with neurons. These are not capable of conducting action potentials, for instance. They don't have the correct voltage-gated ion channels for conducting action potentials, but these processes are still known as dendrites. Okay, so here we have one of these dendritic cells. So the cell body is underneath the surface of the alveoli. So these are not actually inside the alveolar spaces in the same way that the alveolar macrophages are. However, these are resident cells that are present within the lung, and they have these little processes extending through the pneumocyte epithelium here. And they are also a phagocyte which will be capable of phagocytosing the Mycobacterium tuberculosis. So if their processes came in contact with a Mycobacterium tuberculosis cell, it would then get phagocytosed up into the dendritic cell. So remember, the Mycobacterium tuberculosis cell will be tiny compared to these human cells. So you would be able to phagocytose it into one of these processes. Okay, so you have these dendritic cells then that are also resident cells within the lung, and they are very, very good at antigen presentation. So, where have we got to so far? So we've got these macrophages and dendritic cells. These are going to be phagocytosing Mycobacterium tuberculosis. And by the way, the dendritic cells, they're also quite bad at phagocytosing Mycobacterium tuberculosis. Everything's bad at phagocytosing Mycobacterium tuberculosis. Often the cells will get the better of all of these different types of phagocytes and will end up breeding inside of the phagocyte and then breaking the phagocyte open and spreading elsewhere. Okay? However, occasionally macrophages or dendritic cells will get the better of the Mycobacterium tuberculosis cell, and they will succeed in killing it. They will break it down, and they'll break its antigens down into loads of little fragments, and they will then present these fragments attached to proteins on their surface known as major histocompatibility complex class II proteins. So I'm going to show this in the context of a macrophage, simply because macrophages are easier to draw than dendritic cells because they don't have the dendrites. I can just draw a great big blob of a cell here. So here is my macrophage, and now on the surface of the macrophage we have protein complexes known as major histocompatibility complexes class II. So this is the MHC class II protein complex. So this stands for major, that's the M, histocompatibility, that's the H. So all one great big word here, histocompatibility, and then complex, that's the C, and then it's class II, which is the -2. Okay, so on the surface of antigen presenting cells, which are these cells that are going to be capable of activating T cells, activating the adaptive immune response, we have these major histocompatibility complex class II proteins, and you have absolutely loads of them. This is completely out of scale. They would be tiny in comparison to the cell. They're tiny little protein complexes on the surface of the cell, and you'll have a huge number of them all over the cell. And the special thing that these protein complexes are capable of doing is they're capable of having little fragments of antigens put in a special groove on the surface of the protein complex known as the peptide binding groove. So these protein complexes have a little groove on their surface known as the peptide binding groove, and once they have chopped up a pathogen, they've digested the pathogen, so once the antigen presenting cell has got the better of the Mycobacterium tuberculosis and it's broken it down, it breaks down its antigens into little fragments, peptide fragments. It can put these peptide fragments on its MHC class II proteins. Okay? And generally, the peptide fragments that are put on the surface of MHC class II proteins will be between 12 and 20 amino acids. So alpha-alpha just means amino acids. So here in green, that little line is supposed to represent a fragment of one of these peptides. A peptide fragment of one of these antigens. And by the way, an abbreviation for antigen is AG. So just to draw this in here, here is the phagosome, and we digested our Mycobacterium tuberculosis. So here it was digested, its proteins were broken up into little fragments, and loads of these little fragments were put on MHC class II protein complexes on the surface of the antigen presenting cell. So I want to stress this because this is important. The Mycobacterium tuberculosis, it will have had a huge number of different antigens. Okay, each of those antigens will have had loads of possible peptide fragments that could have been produced by splitting it up. So each of the antigens, you will probably possess loads of them. So the Mycobacterium tuberculosis, it's unlikely to just have one copy of a certain antigen. It's probably got hundreds, if not thousands, of copies of that antigen. They're all going to be chopped up in different ways, so you'll produce loads of different peptide fragments from a single antigen. And all of these different peptide fragments from different antigens or from the same antigen, they're all going to be put and displayed on the surface of the antigen presenting cell on different MHC class II protein complexes. So the antigen presenting cell is now absolutely covered with different peptide fragments associated with the Mycobacterium tuberculosis, and it could use any one of these peptide fragments to initiate an adaptive immune response. Okay, so this Mycobacterium tuberculosis cell, do you think it's just going to initiate one adaptive immune response where you just get one type of T cell being activated? No, instead you get absolutely loads of different adaptive immune responses initiated against loads of different examples of the different peptide fragments that we've now got on the surface of this antigen presenting cell. So a single pathogen such as Mycobacterium tuberculosis can get loads of different examples of the same adaptive immune response initiated against it, where the different adaptive immune responses are targeting different peptide fragments. So the different T cells that you're activating will be targeted against the different fragments from potentially the same antigen or different antigens of the Mycobacterium tuberculosis. So I hope that's clear. Okay, so we've got our antigen presenting cell that now is covered in these peptide fragments from antigens of the Mycobacterium tuberculosis. It now needs to go and present what it's got to T cells of the adaptive immune response to try and activate them. But before I just discuss that, I want to discuss the fact that this antigen-presenting cell is also going to put co-stimulatory molecules on its surface which are going to be important in activating the T cells of the adaptive immune system. Okay, without these co-stimulatory molecules, it won't work. So, the antigen-presenting cell that has come into contact with the pathogen will have had all sorts of pattern recognition receptors activated on its surface, and these pattern recognition receptors would have activated all sorts of signaling pathways inside the cell that would have led to the phagocytosis of the pathogen, for instance, and the production of more pro-inflammatory cytokines such as interleukin-1 and TNF-alpha to maintain the inflammatory response. But another thing that those intracellular signaling cascades downstream of pattern recognition receptors result in is the expression of certain molecules that are really important in activating T cells on these antigen-presenting cells. And these molecules are known as B7 molecules, and I've drawn one here. So I'll just colour this in orange. So B7 molecules, of which there are two major examples, B7.1 and B7.2, they are absolutely essential if this antigen-presenting cell is going to succeed in activating T cells. And the antigen-presenting cells do not usually express B7 molecules. They are known as co-stimulatory molecules. And as I say, they're going to be absolutely essential for a protective mechanism to make sure that we're not getting the activation of T cells in response to auto-antigens, in response to peptide fragments from self-antigens, auto-antigens. Auto, remember, means self. So we'll see how this all comes together to be part of a protective mechanism later on. But for now, just know that when pattern recognition receptors are activated, so let's say this is a pattern recognition receptor here, and I'll just colour it in green. So when pattern recognition receptors are activated on the surface of the antigen-presenting cell, and of course they will be activated in this case because it's Mycobacterium tuberculosis, that will cause changes in gene expression, and that will result in the expression of these B7 molecules, which are now going to allow the antigen-presenting cell to successfully activate T cells. So let's now actually continue the story on. How is it going to activate T cells? Well, it needs to go somewhere where T cells are actually present. Now, where do T cells hang out? Well, there's a lot of T cells in the blood. It's not going to find them there. Instead, where it's going to go is the lymph nodes. There are a huge number of lymphocytes, in particular T lymphocytes, in the lymph nodes, and that is where our antigen-presenting cell, either a macrophage or a dendritic cell, is going to go to find T cells that can potentially activate. So how is it going to get to a lymph node? Well, lymph vessels, lymphatic vessels, which drain all tissues of the body, they eventually lead to lymph nodes if you follow them for long enough. So what's now going to happen then is our antigen-presenting cell is going to go into a lymphatic vessel. So I'll just draw this here. So we'll have a lymphatic vessel here, draining this portion of the lung that is currently affected by the Mycobacterium tuberculosis. And what will gradually happen is it will drain into bigger and bigger lymphatic vessels. So here we can have lots of lymphatic vessels joining together to make a big lymphatic vessel. And then lots of big lymphatic vessels will converge together to drain into a lymph node, and the lymph nodes that drain the lung are generally present in the mediastinum, which is the space in between the two lungs. Okay, in the middle of your body, behind your sternum. Okay, so I'll draw now a lymph node here. So here are the afferent lymph vessels, the vessels that are draining into the lymph node. And this will be a mediastinal lymph node that I'm drawing here. So these are all afferent lymphatic vessels, and then we'll have one lymphatic vessel coming out. Okay, so let me just write a few things down. So this lymph node that I'm drawing in this picture, this is a mediastinal lymph node, which will be draining lymphatic vessels coming from the lung. Okay, which of course this is. All of these lymphatic vessels that are coming into the lymph node, they are known as the afferent lymphatic vessels. So I'll colour the afferent lymphatic vessels in orange here. So these are the afferent lymphatic vessels, and they are bringing lymph from lung tissue into this mediastinal lymph node. Now, where is the lymph that these afferent lymphatic vessels are bringing in going to actually go? Well, there is a sinus around the edge of the lymph node known as the marginal sinus, where all of this lymph can drain into. So I'm just drawing this on here. So colouring this in orange here, this is what is known as the marginal lymph node. So just adding a few annotations on here. So these are the afferent lymphatic vessels, and I'll shorten lymphatic down to lymph. So afferent lymph vessels, and then this sinus around the edge of the lymph node, which all of the lymph from the afferent lymph vessels is draining into, is known as the marginal sinus. Now, just to go back a little bit, the outer portion of any lymph node is made up of a capsule. So let me just surround the entire thing by a capsule here in green. So this is a tough capsule surrounding the entire lymph node, and then underneath the capsule, around the edge, as we can see here, we have the marginal sinus, where all of the lymph coming in through the afferent lymphatic vessels is going to go. Okay, right. Now to talk about the one efferent lymph vessel. So afferent means coming into, efferent means going out of. So we have a single efferent lymph vessel here, which will be draining the lymph fluid out of this lymph node, and it will be a bigger vessel than these afferent lymph vessels. What will then happen is this efferent lymph vessel, it might converge with another bunch of very large lymphatic vessels on another lymph node, and then eventually the drainage system will take it into one of the major lymphatic vessels, either the thoracic duct or the right lymphatic duct, which will drain into the venous system. But I'll talk more about that later on. Okay, so here is the efferent lymph vessel, here in red, and this collects fluid from the medullary sinus, which is right in the centre of the lymph node. So this space here, this is what's called the medullary sinus of the lymph node, and this is where lymph collects after it's moved from the marginal sinus through all of this tissue here, which we'll discuss in a moment, and has come into this medullary sinus. Right, so now we've got enough on this picture to actually discuss what these lymph nodes are going to do. What do lymph nodes actually do in the human body? What is the purpose of lymph nodes? Okay, so this is the basic idea of a lymph node. The lymph is going to come in from the afferent lymph vessels. In order to actually get into the efferent lymph vessel, it has to move from the marginal sinus into the medullary sinus. And in order to move from the marginal sinus to the medullary sinus, it has to get through all of this tissue of the lymph node here, the solid tissue of the lymph node. And this solid tissue of the lymph node, this is where all of the lymphocytes are going to be present. So in that solid tissue, you're going to have loads of lymphocytes. Now, there are two different types of lymphocytes. There are B lymphocytes and there are T lymphocytes, often just known as B cells and T cells, and they're both really important in adaptive immune responses. For our purposes, we're not going to be discussing B lymphocytes. The T helper 1 mediated adaptive immune response doesn't involve B lymphocytes, it just involves T lymphocytes. Now, All we need to know about these at the moment is that they are really important parts of the adaptive immune system. Very, very powerful cells for dealing with pathogens. Okay? The basic idea of a lymph node is that it's a site of immune surveillance. Think about this. We are bringing in lymph, which is effectively tissue fluid from loads of tissues, from the lung in this case, but at other parts of the body it'll be from the nearby tissues. And we are making it filter through all of these cells of the immune system, and these cells of the immune system are continually going to be on the lookout for things that are abnormal in this lymph, abnormal in this tissue fluid that is coming from our tissues. And if they find something that's abnormal there, they will launch an adaptive immune response, is the idea. So that's the purpose of these lymph nodes. They are present for immune surveillance. They are monitoring the lymph that is coming from the peripheral tissues for signs of infection. And this is where these antigen-presenting cells are going to present the antigen fragments that they have on MHC class II. So, let me just complete our discussion of the structure of a lymph node with the structure of this solid material of the lymph node. So we can divide the solid material of a lymph node into two different portions. Okay, the outer portion is known as the cortex. So this portion of the solid tissue is the cortex, and then the inner portion here, this is known as the paracortex. So let me try and squeeze this in here. So it should all be one big word, but because of the lack of space I've had to put it on two separate lines. So that says paracortex. So we have the cortex and the paracortex. Now what's the difference between the cortex and the paracortex? Well, in the cortex of the solid material of a lymph node, you're going to have B cell follicles. So let me just draw a few of these in here. So the B cells that are present within lymph nodes, they cluster in these spheres known as B cell follicles. So these structures that I'm now colouring in red, these are representing B cell follicles. These are spheres of B cells in the lymph node solid material. So B cells cluster in B cell follicles, which are in the cortex of the lymph nodes. All of the material surrounding the B cell follicles, that is made up of T cells. So all of the rest of this space, so all of the rest of the cortex and all of the paracortex, this just consists of loads and loads of T cells packed together. Okay, so that's the difference between the cortex and the paracortex. The cortex has T cell material with these B cell follicles interspersed in the T cell material, whereas the paracortex just contains T cells. Okay, so that's the structure of a lymph node and also some insight into what the function of a lymph node is in this immune surveillance role. Now let's talk about what the antigen-presenting cell is going to do. Let's go back to our story. So the antigen-presenting cell has phagocytosed the Mycobacterium tuberculosis over at the site of infection, somewhere within the lung. What's happened then is it's chopped up the antigens of the Mycobacterium tuberculosis and presented fragments of these on MHC class II protein complexes on the cell surface. So we've now got our antigen-presenting cell coated in MHC class II protein complexes with peptide fragments, loads of different peptide fragments presented in their peptide binding grooves. The antigen-presenting cell has also put the co-stimulatory molecules, either B7.1 or B7.2, they both perform the same role and are therefore just known as B7 molecules, on their surface, and is ready now to activate T cells. It is now drained through lymphatic vessels and has got to one of these mediastinal lymph nodes. It's come into the marginal sinus of this mediastinal lymph node, and it's now going to percolate through the solid material of this mediastinal lymph node, and it's therefore going to come in contact with a huge number of different T cells. Forget B cells, we're not interested in those as part of this T helper 1 mediated adaptive immune response. We're going to be interested in T cells. So, the question now comes, which T cells is it going to activate? Okay, so I'll go over the page and let me discuss T cells briefly. So, you have a huge number of different T cells in the lymph nodes of your body and in the bloodstream of your body. And these T cells come in two major types. And these two major types are the CD4 positive T cells, which are also known as the helper T cells, which we can abbreviate down to TH cells. So in full, this means helper T cells. Okay? So this is one of the great populations of T cells. The other major type of T cells is the CD8 positive T cells, which are also known as the cytotoxic T cells. And this is the type of T cell that we're not going to be incredibly interested in here. So the other type, just for reference, is the CD8 positive T cells, which are also known as the cytotoxic T cells, and for short these are abbreviated to TC cells. So this C here stands for cytotoxic. So there are these two major populations then of T cells, CD4 positive T cells and CD8 positive T cells. For our purposes, we are not interested in the CD8 positive T cells. In order for an antigen presenting cell to activate CD8 positive T cells, it needs to have presented antigen fragments on MHC class 2 protein complexes. Sorry, not MHC class 2 protein complexes, the exact wrong one in fact. MHC class 1 protein complexes. And generally, in order to present antigen fragments of a pathogen on MHC class 1 protein complexes, that pathogen must be intracellular and it must be in the cytoplasm. You might say, but Mycobacterium tuberculosis is sort of intracellular, it's living inside these phagosomes, but that's not truly intracellular. In order to be truly intracellular, you have to be inside the cytoplasm of the cell. So for instance, viruses are a good example of a truly intracellular pathogen. Those sort of pathogens, they have their antigen fragments presented on MHC class 1 protein complexes on the antigen presenting cell, and those antigen fragments will be presented and activate CD8 positive or cytotoxic T cells. When we have a pathogen's antigen fragments presented on MHC class 2 protein complexes, then we are going to present them to CD4 positive or helper T cells. Okay, so we're only interested in the helper T cells. Now, helper T cells and cytotoxic T cells, they're not segregated. So in the T cell tissue within lymph nodes, you'll have cytotoxic and helper T cells interdispersed everywhere. Okay, so the antigen presenting cell with these peptide fragments from Mycobacterium tuberculosis on MHC class 2 on MHC class 2 protein complexes is only going to be interested in activating the CD4 positive or helper T cells. Now, the next thing to say is that in this population of CD4 positive or helper T cells, there is a huge amount of variation. So you'll have a massive great population of helper T cells, and all of these different helper T cells, they are directed against different peptide fragments. Okay, so they can recognize and be activated by different peptide fragments, and the job of this antigen presenting cell is now to try and find helper T cells that are targeted against one of the peptide fragments that it has presented on its surface on MHC class 2 protein molecules. So what I just want to briefly discuss is what is it that actually gives a T cell, in our case we're specifically interested in helper T cells, but the same is true for the cytotoxic T cells. What is it that gives a T cell its specificity to a specific peptide fragment? Well, T cells have a receptor on their surface which is called the Tcell receptor, which for short is abbreviated down to the TCR. So all of these helper T cells that we're interested in activating, they're all going to have T cell receptors on their surface, and the T cell receptor is capable of binding to an MHC class 2 peptide complex. Okay, and the peptide in the peptide binding groove of the MHC class II has to be a specific peptide in order for the T-cell receptor to bind. So, CD8 positive cytotoxic T cells, they also have T-cell receptors, but their T-cell receptors will bind to MHC class I protein complexes with a peptide fragment in, rather than MHC class II protein complexes with a peptide fragment in. So let me now discuss what's going to happen. So what's going to happen is the antigen presenting cell comes into the lymph node, it percolates through the solid material of the lymph node, and it's going to come into contact with a huge number of different CD4 positive T cells, which will all have a different type of T cell receptor on their surface. And I should stress that each one of the T cells will only produce a single type of T cell receptor. So they'll have loads of this T cell receptor molecule all over the surface, but all of their T cell receptors will be identical. So all of the T cell receptors on a single cell will be identical, but between different T cells you'll have a different design of T cell receptors. So I hope that's clear. So what now needs to happen is the antigen presenting cell needs to find helper T cells that have T cell receptor designs that can bind to one of the MHC class II protein complexes with one of these peptide fragments that the antigen presenting cell has on its surface. So let's say that our antigen presenting cell does happen to find a match for one of its MHC class II peptide complexes. Let's now draw what's going to happen. So we'll have our antigen presenting cell here, and I'll just straighten this up. Right, so here is the antigen presenting cell, here is its nucleus here, and we know that on its surface it will have loads of these MHC class II protein complexes with peptide fragments of antigens of Mycobacterium tuberculosis in their peptide binding grooves. So let's just colour this in. So this is an MHC class II protein complex here in red. So that's MHC class II. And then in the peptide binding groove of this MHC class II, you've got some fragment from one of the antigens of Mycobacterium tuberculosis, and I'll just colour that in green here. Okay, remember we also have the B7 molecules, but we're not going to see the significance of those for a little while, so I'll come back to those in a moment. So now, let's say that this antigen presenting cell happens to be able to find a T cell, which will have to be a CD4 positive T cell, that actually had a T cell receptor design that was a match for this MHC class II peptide fragment complex. What will then be able to happen is the T cell receptor molecules on the surface of this T cell will be able to bind to this MHC class II peptide fragment complex, like so. So this protein here is the T cell receptor. I'll just give this T cell a nucleus, like so. Okay, right, so let's just colour in the T cell receptor in blue here. Okay, so in addition to the T cell receptor binding to the MHC class II protein complex, what's also going to happen is the CD4 molecules on the surface of these CD4 positive T cells are also going to bind to MHC class II. So I'll just draw this here. So this molecule here, this is CD4. Now CD4 binds to a portion of MHC class II that is on the side of the MHC class II protein complex, i.e. it binds to a portion that is away from the peptide binding groove. So the binding of CD4 to MHC class II doesn't depend on what peptide fragment is in the peptide binding groove. Okay, right, and this is the reason that these antigen presenting cells with peptide fragments presented on MHC class II protein complexes are only going to be able to activate CD4 positive T cells, because In order for the T cell to be activated, not only does the T cell receptor have to bind to the MHC class II peptide fragment complex, but also the CD4 molecule has to bind to the MHC complex. And CD4 binds to MHC class II, whereas CD8 binds to MHC class I. So these ones, because they have CD8, they can only be activated by peptide fragments presented on MHC class I, whereas these ones, because they have CD4, they can only be activated by peptide fragments presented on MHC class II, which indeed we are going to do in the case of Mycobacterium tuberculosis. Okay, so just to complete this picture up then, this is our antigen presenting cell. I'll remind you that the antigen presenting cell had a huge, great plethora of different peptide fragments from different antigens, or potentially from the same antigens, of Mycobacterium tuberculosis, and it was just looking for a single T cell that had a T cell receptor design that could bind to one of these MHC peptide fragment complexes, and that's what it's found here. So over here, this is the CD4 positive T cell. And I might just add in front of that name naive, because it has never been activated before. So this is a naive CD4 positive T cell, which means that it has never been activated before. Okay, right, so this complex that we're forming here between the antigen presenting cell and the naive CD4 positive T cell, this is an example of what's called an immune synapse. So this interface between these two cells that is held together by an MHC class II peptide complex binding to a T cell receptor and one of these co-receptors, CD4 or CD8, this is known as an immune synapse. Okay, right, so we form this immune synapse between an antigen presenting cell and one of these naive CD4 positive T cells that has a T cell receptor design specific to this peptide fragment of one of the antigens of Mycobacterium tuberculosis. This is now going to give signal 1 for the activation of this naive CD4 positive T cell. So what's going to now happen is this is going to give signal 1 for activation of this naive CD4 positive T cell. However, the naive CD4 positive T cell does not get fully activated just with signal 1. In fact, if it only receives signal 1, then the exact opposite happens. If you only receive signal 1, it would cause this naive CD4 positive T cell to go into what's called an anergic state. And an anergic state is where it never divides and it will never ever be activated no matter what you do with it. No matter how much stimulation you give to it, it will never ever be activated. Now, why on earth would you want to do that? Well, if you only give signal 1, it suggests that the peptide fragment that you're presenting in the context of MHC class II to this naive CD4 positive T cell is an auto peptide fragment. It's a peptide fragment from an auto antigen, and it suggests therefore that you're about to launch an adaptive immune response against a self-peptide. The reason that it suggests that is that if you have actually got this peptide fragment from the antigen of a pathogen, you should always deliver signal 2 as well as signal 1 to the naive CD4 positive T cell. And the reason is that signal 2 is given by the co-stimulatory molecules. So remember, I said that the antigen presenting cell will be activated to express on its surface B7 molecules, i.e. either B7.1 or B7.2, most likely both of them. So it'll put all sorts of B7 molecules on its surface, and these B7 molecules, these bind to a receptor on the surface of the naive CD4 positive T cell, and they deliver signal 2, and it's necessary to have both signal 1 and signal 2 delivered to the naive CD4 positive T cell to get proper activation of the naive CD4 positive T cell. So let me now draw on the receptor for B7 here. So the receptor for B7 is a protein known as CD28 on the surface of the naive CD4 positive T cell. So I'll just draw this in blue. Okay, so to go over the story and put everything together here, if the antigen presenting cell is actually presenting a peptide fragment that it got from an antigen of a pathogen, then it should also have B7 molecules on its surface, which it would only express if pattern recognition receptors of that antigen presenting cell have been activated, and that suggests that the antigen presenting cell has actually come into contact with a pathogen. Okay, oh, and let me just complete this picture up. So this delivers signal 2 here. So if you have indeed got this antigen fragment from a pathogen, then you will have the co-stimulatory molecules on your surface, and you will deliver both signal 1 and signal 2 concurrently, and then this naive CD4 positive T cell will actually continue the process of activation, which I'll discuss in a moment. If you got this peptide fragment from a self-peptide, however, then the antigen presenting cell will not have come into contact with a pathogen, therefore its pattern recognition receptors will not have been activated, therefore it won't be expressing co-stimulatory molecules, and therefore it will only deliver signal 1 to the naive CD4 positive T cell. That's why if you only receive signal 1, it sends the naive CD4 positive T cell into an anergic state, because it suggests that the peptide fragment that this naive CD4 positive T cell recognizes is from a self-peptide, i.e. that this T cell is autoreactive. So this is a protection mechanism to try and prevent autoimmune disease. Okay, right, so enough of discussing this protection mechanism, let's just now go back to the story. So our antigen presenting cell that was covered in peptide fragments from the antigens of Mycobacterium tuberculosis presented on MHC class II protein complexes with co-stimulatory molecules on its surface has gone to the lymph node, and there it has managed to find a naive CD4 positive T cell that has a T cell receptor design that is perfect for binding to one of these MHC class II peptide fragment complexes, and it just needs to find one. Okay, it might find multiple and might therefore be able to activate multiple of these concurrent adaptive immune responses, and indeed different antigen presenting cells might activate different naive CD4 positive T cells with different designs of T cell receptors, and therefore we'll get lots of concurrent adaptive immune responses being initiated that will all result in similar outcomes but directed against different peptide fragments of the antigens of Mycobacterium tuberculosis. But we're just focusing on the story for one of these peptide fragments now to keep it simple. Okay, so it's now delivered signal 1 and signal 2 then to this naive CD4 positive T cell. What now is the process that this naive CD4 positive T cell is going to go through in order to complete its activation? Well, to complete its activation, it now needs to receive signal 3, and let me just discuss what signal 3 actually involves. So, when you get signal 1 and signal 2, it changes gene expression within the naive CD4 positive T cell. In particular, the naive CD4 positive T cell starts to produce a lot of interleukin-2 proteins, and also a component of the interleukin-2 receptor known as the interleukin-2 receptor alpha component. Okay, so let me go over this. So let's say we can zoom in on a little piece of membrane of this naive CD4 positive T cell. So let's take that little box that I've drawn here and zoom in on that. Okay, so here is a portion of the membrane of our naive CD4 positive T cell. Now, before it had received signal 1 and signal 2, the cell expressed two components of the interleukin-2 receptor, which are known as the interleukin-2 receptor beta component and the interleukin-2 receptor gamma component. And actually, I should take a step back. The interleukin-2 receptor is the receptor on the cell surface membrane for interleukin-2, and it's a trimer. It's made out of 3 separate pieces, and the 3 separate pieces are given nice simple names: the interleukin-2 receptor beta component, the interleukin-2 receptor gamma component, and then finally the interleukin-2 receptor alpha component. And I've stated those in a slightly odd order, but never mind. The interleukin-2 receptor beta component and the interleukin-2 receptor gamma component, they are normally expressed even on these naive CD4 positive T cells that have never received signal 1 or signal 2. Okay? The interleukin-2 receptor alpha component is not, so that means that the CD4 positive T cells that are not in this naive state usually do not have complete interleukin-2 receptors. When they receive signal 1 and signal 2, they're going to make the final component of the interleukin-2 receptor, which is the alpha component. This is going to be trafficked to the cell membrane, and it can then bind with the interleukin-2 receptor beta and interleukin-2 receptor gamma components to make the full receptor. Okay, so this is what I'm drawing here. So the interleukin-2 receptor gamma component will be trafficked to the cell membrane, and then it combined with the interleukin-2 receptor beta and gamma components, which were already pre-produced, to create us a full interleukin-2 receptor. The cell will also start producing interleukin-2, which will secrete from its cytoplasm into the extracellular fluid, and now the interleukin-2 will act on its own interleukin-2 receptors. So this is actually an example of autocrine signaling. So when a cell does something like this, when it secretes a molecule that's going to act on its own receptors, that's known as autocrine signaling. And this is what this T cell is going to do in order to complete the final stage in its activation, which is signal 3. It's going to produce the receptor, the final component of the receptor, and produce the ligand for that receptor. And once the interleukin-2 has stimulated the interleukin-2 receptor, that will activate another intracellular signaling cascade, which is now known as signal 3. And signal 3 will then cause full activation of this naive CD4 positive T cell. And we'll have a break here, and we'll discuss what signal 3 is going to do to this naive CD4 positive T cell in the next video.